Sure.
Thank you.
I am Donna Santillan. I am a research professor at the University of Iowa. I'm the vice chair of research in the Department of Obstetrics and Gynecology and the division director for reproductive health sciences. I've been at the university for 18, 19 years now. My PhD is in molecular biology. And I studied leukemia originally in graduate school. But when I was bored, what was laying around our house was Mark's magazine, Mark's journal. And I started flipping through them and couldn't believe the very basic things that were being published that I thought we should know already about women's health and about pregnancy, because women have been getting pregnant for a very long time. And so you would think that some of the things have been worked out already and studied. And it's sad and it makes me angry to know that they haven't, because people didn't really care about including women in research for a really long time, and really not until very recently, as we'll talk about. And so that is sort of how I moved into this field. When we came to the University of Iowa, I interviewed and did a postdoc in the Department of OB-GYN and then stayed here. And so we were kind of buy one, get one free for the university. I think I'm the free one. But so we have our own research programs, but we do collaborate a lot, and we do some similar work. And so today I want to talk about the importance of maternal health research and some of the tools that we've developed at the University of Iowa to help speed up the work, because it has lagged behind for so many years. And I'm sure I'm preaching to the choir that it's important to study maternal health research, but just to give you some of the background on how it's been understudied. There we go. So maternal health is very understudied, but before even that, as a subcategory, women's health is understudied. It wasn't until 1993, and I will age myself by saying I was in high school when this happened, when the NIH, the government part where we get our big dollars for grants, said that you actually have to include women in research. 2 scientists, right? That is not that long ago, right? That's 31 years ago. Just that we said, if you want NIH money, you have to include women or justify why you're not. And so that's sad. And it wasn't until 2014, 10 years ago, when applications, besides if they were about humans, if you were going to be using animals in your work, or if you were going to be using cell culture cells, right? Like you've heard of HeLa cells and Henrietta Lacks, like derived cells like that for research, that you had to talk about, were those females? And justify that. And you couldn't just say we're only using male mice because females have those messy hormones. I think people probably still tried to say that, but they began to make that be part of the application and considered. And it wasn't until 2018, 6 years ago, when they removed the common rule, that this vulnerable designation from the pregnant population, which meant that pregnant women had the same rights as a child or prisoner for deciding if they wanted to participate in research, right? So this is how you haven't even been included, right? And we just took research from men often. Well, we didn't do it. People, the researchers in the past took research from men and tried to apply it to pregnant women, and that just doesn't always work very well. And if you were looking back at women's health research, they did a study to look at what have been barriers to progress and what needs to change to improve research. And so in this report, they said there's been inadequate attention to social determinants of health, a lack of attention to environmental factors in women's health research, inadequate enforcement of including women in studies. So they didn't say it until that long ago that they had to be in them, and then they didn't even enforce it when they started saying it, right? And a lack of accounting for sex and gender differences in results. So they would just put everybody together, pool the results, and not see, were there different results for women? And then a poor communication of results. And we saw that with COVID in general, that scientists don't always do a great job of communicating about science to the public, but especially about women's health research as well. So the National Academy of Science made some recommendations on how this can be improved. This including, if we wanted to improve women's health research in general, not only just pregnancy related, that it had to be research across the spectrum of research. all the kinds of studies really need to be improved. So if it's cellular, animal, molecular, observational studies, clinical studies, the implementation work like Elisa talked about this morning, Dr. Faro, like all of that needs to be improved for women's health and maternal health. It's not just one area that is lagging behind, it is actually all of it. We need to increase research on populations of women that have the highest risk and the highest disease burdens. We need to promote wellness and quality of life in addition to mortality. So, there's lots of emphasis, if you look back at old papers, on mortalities and morbidities, but, it's not just about death or having very bad outcomes. There are other things that contribute to poor quality of life, and trying to keep people healthy and not just stopping people from being sick that need to be studied. We need to study common determinants and risk factors that underlie multiple diseases. We need to look at pathways and behaviors. I already mentioned the sex stratified analyses, so sorting out males and females and the differences in results. And then doing the kind of work that Dr. Faro talked about this morning with implementation research and making sure that it gets implemented and is used in a way that actually works in society, and improving the communications of research findings, because you want to reduce confusion over conflicting results. And you saw a lot of that in COVID. Research changes, as you talked about earlier this morning as well. And so it's not that it's always wrong or it's conflicting. We get better at it, and assays change and studies get replicated. And so it's just there's an evolution of our knowledge, and we have to make that clear to people and when things were wrong or not done right or what the difference is. And so, if you take a small study, which should be done, and you test it in a very small group of people and you see a good finding with 10 people, but then you go on and do the bigger study with 1,000 people, it might not pan out. But if someone reads the paper about the 10 people, well, why are these different? And understanding that with 10 people, you could just get random differences that don't hold up in a bigger group. And so trying to explain that to people and why you can't just take the results of some studies and apply them. So we need to do better education. And I don't have to ask this question here, but when Mark and I give talks, we often will say, how many people here have been through a pregnancy? And ask them to raise their hands. And then, when we give public talks. And so you'll get most women, not all, but some women will raise their hand. And then you get like a random guy who will raise their hand. Somebody like Mark or Tom will raise their hand, and they get kind of laughed at, usually by the crowd, but they're not wrong, right? Because last time I checked, every single person is born. And so that's part of the education and telling the stories that make it important. We aren't just talking about women's health, and we aren't even just talking about the effects to people who have had a pregnancy in their health. Pregnancy really affects every single person's health on the planet because we're all born. And Mark will talk about after me the effect of pregnancy and specifically preeclampsia on not only the birthing person's long-term health, short and long-term health, but also on the child's health. Right. And so this really is a general problem for society, and we have to make people realize that this is not just a women's health issue. Everybody should actually care about it, and we should actually put resources into studying it. And so there's just another way that we show that everybody comes from a pregnancy. And sometimes I show a slide that has Wilma Flintstone giving birth to Pebbles, just to show that women have been giving birth for a long time. And that there's just so much that we don't understand and don't appreciate yet. And the sad thing is that pregnancy is really understudied. Given that pregnancy has been going on for a really long time, if you look in PubMed, which is where scientists look for papers, it's like Googling, but specifically for papers, and you look up cancer, which is very important to study, but it is not something that affects everybody. So if you put in the term cancer, you will get over 5 million papers have been published about cancer. But if you put in the word pregnancy, you get 1.1 million. And as I said, pregnancy affects everybody, but it's 1/5 of the papers, right? Just compared to cancer, not to everything else. And so that's just kind of sad and demonstrates why we need to increase the amount of pregnancy-related research that is going on. And like I said, it is not just a women's health disease. It's not just a pregnant person's disease or a pregnant person who's gone through preeclampsia. Pregnancy really affects, and the conditions of pregnancy really affect your long-term health, and it affects the child's health and even the grandchild's health. And so there's a lot of diseases that have been associated in multiple generations going backwards to somebody and how their health was in pregnancy, right? So if I think about my grandmother, honestly, she lived to 96, but it was not healthy living that got my grandma there. She smoked like a chimney. She drank a lot. She told me to smoke when I was pregnant so that the baby would be smaller and it would be easier to deliver. Do not follow that advice. I did not. But if you think about that my grandmother did that, then that is part of the explanation for maybe why all of my mom and my aunts have asthma. And then when you think about that when my mom was in utero, within my grandma, well, I was one of the oocytes and the eggs in my mom that eventually became me. So my grandmother smoking has multi-generational effects. And so when we improve one person's health, we're improving the health of multiple generations. And so that's really powerful in pregnancy-related research, that we can not only affect one person's health, but we can affect multiple generations. And you need this research to not only just to understand what's going on, but we need to improve clinical care now and in the future, and for many reasons. And so I know earlier, Mark and Tom were talking about rural healthcare and some of the differences and different problems that we face. And so if you look at this map of Iowa, on the left in 2000, Iowa has 99 counties. All the counties in yellow had labor and delivery services, and the ones in red did not. And if you look to 2019, look at how many more counties do not have labor and delivery services. And this is actually far worse now. So it's 32 counties now that have labor and delivery services out of the 99 in Iowa. So 17 more have closed since 2019. And babies don't always come when they want, and moms don't always have a lot of time when they're sick. And so if you are in the middle of some of these maternity care deserts, you don't have a lot of time to get to the cities that have the higher resources for the mom or the baby. And so it's really important that we do research so that we have better biomarkers and we have better tests and we know who's at higher risk and really needs to be seen somewhere else. And so earlier when you were talking about who should go see MFM or not, maybe if we had some of these screening tests, we could say, well, you live in a very rural area and your screening tests were really high, you need to go see MFM or not, and then not put a burden on the resources, but send the appropriate people for the care. And also not sending everybody 2.5 hours to go talk to somebody and find out they're probably going to be fine or not know, so that you can really improve life. So one of the ways that we have worked to improve knowledge around pregnancy is biobanking. And we have a pretty unique resource. I know that others have biobanks, but I think what we have developed at the University of Iowa is fairly unique in that ours really spans all of the lifespan. So ours is called the Women's Health Tissue Repository. I am the director of it. Mark is the clinical director. We consent patients at the University of Iowa Healthcare to participate, and we've tried to make it very easy to participate in this. We collect extras alongside clinically indicated samples. So when you're pregnant, you have to give a lot of urine samples, and there's a lot of different blood draws. And so when you do that, we just get an extra tube of blood. So there's no extra needle sticks. When they use your urine in the back, the sample goes off for what needs to be done clinically, and then one of my lucky people in lab gets to take the urine cup and pour the extra, and we store it for research when people have consented for that. We code these samples to protect patient privacy, so it's unrelated to the medical record number. And then when we have these when other researchers have approvals, they can obtain samples from us and get coded clinical data. So we can protect your privacy, but we can make it really easy for people to do research fast, right? Because, you hope pregnancy lasts as close to 40 weeks as possible. Some people have shorter ones, obviously, for reasons. But you don't want to have everybody having to go consent different patients, see who might develop preeclampsia or whatever they're trying to study, and then waiting the amount of time. So it just speeds it up to go to our freezer, pull out the samples, and start your work. We also ask patients if they want to join a registry for future studies, because there's a lot of other work going on that some can use our samples, but then there might be other follow-up that they want to do 1 or 2 years later or multiple years later. And so then we have this registry that can be used of people who already said that they were willing to participate in research. And we store all this information. We have this multi-million-dollar software that the university bought where we store information about the people who've agreed to participate, their sample information and their consent information. And just a lot of details are in there so we can keep track of all of it. We work with our tissue procurement core at the university for collecting samples for other parts of the lifespan and pregnancy. We have standard operating procedures, so we know exactly how every tube of blood is handled. And we're not getting leftovers from your clinical. It's always the same kind of tube. So, if you notice when you get blood draws, they have different colors on the tops of the tubes. And it means because there's different stuff on the inside of the tube. And that can cause results of tests to be different. And so this way, we always know every tube is the same and every tube is the same between our banks. And so if we're comparing non-pregnant to pregnant or cancer to not cancer, we know that all the tubes were collected the same, handled the same, stored the same. They're not leftovers that we don't know how long they've been set out. If they were a red top, a blue top, they're all the same. And we have them in freezers that have temperature monitors on them and they're locked up. And they page us and call us if the freezers start to fail so that we can come in and move the samples and save them. So we put a lot of effort into building a huge repository and really being good stewards of the samples that people are trusting us with, because we really do appreciate that people have consented to share their biological samples and their clinical information for a study that is not going to help them. It's really about improving this for other people. And so people really are being very generous, and we want to be good stewards of that. So the Women's Health Tissue Bank actually consists of 4 different biobanks that are each directed to different stages of life. So the Perinatal Family Tissue Bank is our pregnancy biobank, and that has over 13,000 people in it. And that's unique people. There are hundreds of people that are in there multiple times for multiple pregnancies. So we consent the pregnant person, they consent for the baby, we can follow their health long-term at the university. So for the mom, we can follow them forward and backward as long as they're getting care here. And then we can follow the children until they turn 18. And then we have a reproductive endocrinology and infertility tissue and data bank. This is primarily infertility patients, but it can also be people who are being seen for endocrinology issues. It also can be adolescent gynecology is included in this bank. And we have different consents for people facing infertility versus the other issues so that our consent forms are directed to the right population. And again, here we consent the patients, their partners. We also do that for the Perinatal Family Tissue Bank. We can get the partners as well. And then any children that may come from infertility treatments. So we have about 7,000 patients in there. The Well Woman Bank is our benign gynecology bank. In general, this is patients being seen in urogynecology. And so that one is smaller, just not as many samples there. And then our gynecologic malignancy. So this is people who are may or just be suspicious of having gynecologic cancer, such as ovarian cancer, endometrial cancer. And so we get samples from them as well. These 2 banks, the Perinatal Family Tissue Bank, so the Pregnancy Bank and the Well Woman Bank, also have surveys associated with them. So in the Pregnancy Bank, we survey people each trimester and ask them about some of their exposures, some of the socioeconomic determinants of health in that as well. To participate in the bank, you have to be English speaking, over 18, over 14 for the Adolescent Gynecology Bank, not a prisoner, able to provide informed consent. We want you to know what you are agreeing to, and not be known to have HIV or hepatitis C, and that's for the safety of the people processing the samples. We collect samples from the birth parent and the partner, the maternal urine, the umbilical cord blood, which is normally just discarded, and the placenta, which is also normally just discarded. And we keep samples of all of that. From the other banks, from like the REI bank, we can get seminal fluid from the males. We get, if you're undergoing IVF and they're retrieving eggs, we get follicular fluid and the cell pellet. From tumors or potential tumors, we get the tumor and then normal adjacent healthy tissue. And then we get the surveys, and then from all the banks, we also get clinical data. And so we have a lot of freezers that hold these samples, and then when investigators need them, here at the university and other places, we can share them with them. We can just go to the freezers and pull out what they need. They tell us they want people in a specific age range or with a specific diagnosis. It's a lot easier to go do that. And so We have over 7,300 maternal child and health pairs, over 100,000 aliquots. And these are all the different diseases that and conditions that have been used from the perinatal family tissue bank, so the pregnancy biobank. So you can see it's been used to study a lot of things, including preeclampsia, but also things like depression, suicide ideation, group B strep, obesity, COVID, vaping in pregnancy, aspirin use. It's been used for over 150 projects, 30 manuscripts, and contributed to 5 different patents. So we're really hoping that we can increase the speed for research, and Mark will be able to talk more about what he has been doing and as an example of that. But I mentioned the blood, we collect it the same in all the banks, the same tube. We keep the watery part of the blood, the plasma, and we also keep the cells, the white blood cells that are in there. We keep them so that they're viable. We can bring them out. They're still alive. So they're useful for a lot of different types of work. We've really thought about how to do this to make it as useful as possible to different people. Like I said, we keep extra urine. We can also ask women if they're willing to pee in a cup and give us extra urine samples. And for all of these samples, we take and we divide them up into smaller amounts called aliquots, and we freeze them long-term in our freezer. And so it's not only that one sample is good for one experiment, it can be used for multiple different research projects. And then, like I said, we do the follicular fluid, umbilical cord blood, placenta, and tumor. So we have these protocols that really help us to know how the samples have been treated. But having samples is great and it improves research, but you also need to have the clinical information to go along with it. And that's also something that is not well characterized and missing. So some people will use like large insurance data sets, but you can't actually tell what kid goes to what mom if you were trying to look at long-term outcomes. In a lot of data, they kind of make assumptions to link the child to the mom or to and we've worked very hard to make sure that we are accurately linking the samples to the right clinical data and the right birth parent to the right child. Because if you have multiple children, it could be easy to make a mistake. And so we've worked on how to do that. And we've spent a lot of years with our biomedical informatics group here, our computer group, for validating the data, organizing it in a way that makes sense, putting some formulas and algorithms around the data so that it makes a little bit more sense for use. And they've built this web-based viewer to help us do this. And then we've added in some social determinants of health data. There are some codes to tell if you live in a more rural or a more urban area. And so we've also added in things like, what is the distance from your address to OB providers or to hospitals, so that we can look at health outcomes, including that information as well. And then we can also link that to some environmental data for some of the studies that we're doing. So if we know your zip code or your census tract, then we know other things about the area that you live in and what you might be exposed to, and that can be shared with researchers as well. So we have all of these variables that are in there, and the nice thing is when we find another variable and we add it, we're adding it for everybody who wants to use it in the future. And so we keep track of a lot of things about the pregnancy and about the child and the long-term health of the mother and the child. So diagnoses, we keep your OB history, how long the delivery was, how did you deliver, was it a vaginal delivery or a C-section, what kind of anesthesia you had, if you had a C-section, why did you have one, just a whole lot of details, what medications were you taking and things like that. So we have that all readily available, and it can take us minutes to get the data together to answer research questions instead of having to wait weeks for the informatics group to pull it, or for making poor students and grad students and residents and things go through charts and find data. And this is also another way to keep data more private and to gather data quicker from a larger group of people to improve the research. So we're very grateful to the patients and the healthcare team that help us in collecting this data, and especially to the patients who have agreed to participate and to share their data, to the administration for supporting this, and the funders who have helped to make this possible. It's really been important work. And, you know, like Kara, I know some of these families, and I know the Bridgewaters, and I was pregnant at the same time that Shelley was pregnant with Haley. And so I'm always moved by her story and always think about that at the same time, I also had a very different outcome than Shelley when we had similar due dates. And our daughter, March 2, was born in March instead of in January and had a healthy outcome. So I am happy to take any questions if you have them now, or else I can wait until after Mark talks as well. Whatever you want, Kara.
There is a question Lisa asked. So she said that she donated her extra unused embryos after her very severe health case. And I know Lisa personally, she had, I believe, liver rupture and had a lot of other health issues, like significant health issues. So she didn't go on to have any more babies. Could embryos, donated embryos, be, let's see, what is she asking? Could embryos have helps, can those embryos that she donated or offered to donate be helpful to scientists to figure out the underpinnings of preeclampsia or HELLP syndrome?
They could. There are restrictions on the use of embryos in research. So you can't do any federally funded research with embryos. You have to have a different source of the funding to do that work. And again, this, we are different. Like, we are collecting samples at the University of Iowa, and so we use the samples that are here. We do also have patients who have undergone IVF and had extra embryos that choose to donate them to research here as well. We haven't got, but PF is a different group. And so we haven't used those samples, and ours is a little bit different than what they have. And we, so I don't know what they have done with their samples.
So Lisa, it sounds like somebody could probably use those embryos in research. But you could probably follow up with those organizations to figure out if they did or not.
And there's a lot of people who have different collections that they've done for specific projects. And ours is a little bit different in that we approach people very early in pregnancy without caring about what their history has been, and we're not waiting until they have a diagnosis of preeclampsia. By having samples really early in pregnancy, it can help understand what is going on so early, so that you can intervene before sort of the horse is out of the barn. And so you would know it who is at higher risk, and Mark will talk more about how he has done that.
Great. Well, I think we can move on if Mark would like to continue.
Yep. Donna, you want to? Sorry. Technology, no move over here. Let me see if I can get me shared here. Let's see. Okay, what do you guys see? Do you see presenter view or the real slides?
Yeah, click present or whatever the little doodad is at the bottom there. I can see the slide plus all of them on the side.
It's your program.
Oh, really?
Mm-hmm.
Oh, that's weird.
Mm-hmm.
Okay. Sorry. Let me stop sharing there. We've just been living in Zoom for the past umpteen thousand years, and I still don't know how to use it. So
it's okay.
Just have too many monitors is really the problem.
I do have 4 monitors, so that's probably my problem.
That might be part of your problem.
Let's see. Share again.
Yeah, I don't like it when you like open it on one, but like shares the other screen that you have. I have that, always have that problem, and then I'm sharing the presenter view and the notes view instead of what I want to.
Let me try that. And
there you go. Oh, no.
I got you. Ta-da.
Ah, yay.
Okay, good. All right.
All right.
Well, hello, everyone. Thanks again for tuning in. And Donna gave you a great discussion of some of the resources that we've helped develop over the past thousands of years that we've been in Iowa. And here I want to show you at least some of the things that we are doing in the lab to kind of address some of these challenges with regard to preeclampsia. Lisa, I noticed that there was a chat question about are we getting paternal samples? We have some, but not a lot. And the question about smoking too is a really interesting one. Yes, it reduces risk. that's like a 6-week tutorial on the mechanism. Some people think it's sort of a response to what happens with the hypoxia that you get with or the low oxygen that happens after you smoke. So long discussion, but the relationship is real. We really are not sure as to why yet, but don't smoke just to stop preeclampsia.
The
Aspirin is
benefit does not outweigh the risk.
that is correct. Correct. So I don't have to tell this group how important preeclampsia is to study. We have a bunch of cases a year, at least 500,000 cases a year, and then 600,000 moms and babies die every year worldwide due to preeclampsia. So every minute that is clicking by, someone is dying. And part of the problem is that we don't have like the diagnostic test. When I have chest pain and I get an EKG and someone orders a troponin, people will be able to say, yep, Mark's got a diagnostic heart attack. We don't have that in preeclampsia. We don't have, there are some competing animal models that kind of look at different parts of preeclampsia, but not the whole thing. And then treatments are often limited to delivery. And this is in the setting of a disease that is much more common than heart attacks, prostate cancer, and colon cancer. And so I think this highlights some of the things that Donna already said with regard to this kind of lack of focus of research in this field. This is one of my favorite slides. When you look, it's not just the immediate stuff with preeclampsia, it's the long-term stuff. And many of you already know this, that those who have a history of either metabolic syndrome, like diabetes, obesity, and smoking, you may have a 4-fold increase of having future cardiovascular disease. And if you overlay preeclampsia on top of that, particularly kind of the earlier forms of preeclampsia or a more severe form of preeclampsia, that risk can be as high as 10-fold. It's not great for the kiddo as well. So kids born to moms underneath these preeclamptic pregnancies are also at increased risk in the future for stroke, epilepsy, diabetes, and if they're born early, infertility. And so this is, as Donna already mentioned, pregnancy in and of itself, and as a subset of that, preeclampsia affected pregnancies, they affect everyone in that dyad. What I like to also focus on, and people that we've talked a little bit during the day, between this relationship between neurologic and psychiatric problems associated with preeclampsia. This is just some data showing that if you are stressed out 6 months prior to your pregnancy, and stress means something significant, including like loss of a family member, your risk of preeclampsia is 4 times higher. Depression and preeclampsia are a two-way street. If you have depression and no other risk factor, you have a 3 times higher rate of developing preeclampsia. And the flip is the same. If you have preeclampsia, you have a 3 times higher risk of developing depression. We in our lab published that birthing parents who have more than 4 adverse childhood experiences, meaning anything from abuse to divorce to death in the family, if they are affected that early on in their childhood, that they are at 7 times higher risk of developing preeclampsia. So there's that link between this cardiovascular disease of pregnancy and future neurodevelopment as well as neuropsychiatric development. As you can imagine, this is not great for the kiddo as well. Kids born underneath these preeclamptic pregnancies are at increased risk for a whole host of psychopathology like autism, spectrum disorder, schizophrenia, mood, and anxiety disorders. And these pathologies are linked to real changes in the neuroanatomy of these kiddos, where the vasculature is less, and they're at increased risk of cerebral stroke. And all of these changes are also associated with impaired cognitive development in these kiddos. So that as an opening foray into preeclampsia, what my lab's been really focused on is sort of kind of soups to nuts with regard to that. Can I predict it early? And once I get there, can we try to fix it? And then what happens afterwards? And so those are the big questions that we've asked in my lab. And so 10 years ago now, we published this paper looking at copeptin. Copeptin is a marker for something called vasopressin that gets released in our brains in response to stress and in response to blood pressures. And so what we showed that copeptin, which is a marker of vasopressin release, is elevated throughout gestation in anyone who develops preeclampsia. The wow, that's cool moment is the fact that in the first trimester, it is elevated as early as the 6th week of gestation. So kind of think about that sort of the implications of that knowledge early on in pregnancy, particularly in a place like Iowa, where our OB-GYNs are far and few between, that it may actually, at the time of pregnancy, you potentially could say, hey, congratulations, you're pregnant. Oh, and this is as early as the 6th week. Oh, you're at increased risk of developing preeclampsia. Maybe you should not be getting care in this in this facility where they don't have any blood or they don't have any OB-GYNs to help take care of you. Our finding, we were the first to show that this copeptin measurement was elevated in the first trimester, but plenty of papers after us have shown this relationship in being able to predict preeclampsia. That's led to patents and our ability to predict preeclampsia, and we're currently working with some companies in order to commercialize is such a horrible word, because the only way you can get something to the market and for patients to actually use is to go through companies in order to develop the test that they need, either in the hospital or out. So we're working with a company in order to get a pee on the stick test right now. So I am a basic science nerd at heart. And so what the humans are telling us is, hey, vasopressin is really elevated early on in gestation. So it's either a setup for preeclampsia or it's a causative factor. So we made a mouse model where we basically gave vasopressin through a little itty bitty pump in these mice throughout gestation to see what would happen, and we got them pregnant. And what we showed with these mice is that they developed all of the things that we expect human preeclamptics to look like. They had a pregnancy-specific hypertension. The kidneys were messed up with increased protein in the urine, as well as some clear pathologic findings in the kidney. Babies were grew small, and there were clear immunologic changes. And I won't go into the detail of that because that would be another 6-week course. But there was clearly more inflammatory things going on immunologically than anti-inflammatory things. As I mentioned before, this can affect the offspring, and so one of my former postdocs, also now a faculty member here at Iowa, looked at the babies born to these vasopressin preeclamptic mice. Okay, and what they can see, there's a sex-specific problem that we see in females. There is a clear motor problem that we see. This rotor rod is basically this little rod turns very slowly, and then the mice walk on it and balance on it. Females were more likely to fall, so there's a very specific motor deficiency we see. In this Y maze, females had a decreased spontaneous alternation ratio. This is a sign of cognition that people take a look at. And then males specifically spend less time in the open arms. If you look at this elevated plus maze, there are walls here, and then there are open parts of this plus maze. If you don't spend time in the open part of this maze, it's a sign of anxiety. And so we saw that as happening in these pups born to the preeclamptic dams or moms. So why do we even bother trying to replicate this in mice? Well, we can't test everything in humans right away. So we need a proof of concept. So what we did is we took, and this is some of the work that's going on in my lab now, we took these mice and gave them betamethasone. Many of you know what betamethasone is. It's that steroid we give women in pregnancy if they're at risk of delivering early, to help prevent some of the bad things of prematurity, like problems with the lungs and problems with the gut and problems with the liver and the brain. And so we know that works on human babies. What happens if we give the preeclamptic mom mice betamethasone at the time of implantation? And what we could see is that it reverses the preeclampsia phenotype. So by decreasing the blood pressure and decreasing the protein that we see. And I won't go through all of this, but trust me to say that betamethasone flips that the immunology and the inflammation that we see in preeclampsia. Instead of it being very pro-inflammatory, betamethasone decreases that pro-inflammatory state. And at the level of the placenta, part of the reason why we think that preeclampsia happens is that the placenta doesn't work. It doesn't move. And the cells that are supposed to move don't. And so this is what we're seeing. We do a little scratch assay by putting these placental cells together. And then normally if you put a little scratch in a dish, those cells will fill in that scratch. But in AVP mice, if you supplement those cells with AVP, they don't move into that scratch. And we see that we reverse that phenotype. We reverse that dysfunction that we see with betamethasone. So we think that that may be one option in terms of new treatments for preeclampsia. The other kind of potential treatment is a little bit based off of what we think about vasopressin. As I mentioned before, vasopressin goes up to help maintain blood pressures, as well as in states of stress and depression. We know, and what we've shown in our mouse model, was that vasopressin in and of itself during pregnancy can cause preeclampsia. And so what if we have these two things that are increasing AVP? And so we think that prenatal stress is maybe one of the things that helps, and prenatal depression and anxiety may be pushing that preeclampsia phenotype. And so to ask this question, we looked at a whole bunch of data that Donna kind of talked about before. We have that data mart where we can look at 70,000 pregnancies at the University of Iowa. Now, if you were to look in the literature and looked through all the medical journals, they would say that women who take SSRIs, so SSRIs, those are those drugs that we take to help with depression, anxiety, like Zoloft and Paxil. Those are things that we often prescribe to women who are affected by depression and anxiety. There are literature to show that if you take SSRIs, that you're actually at increased risk of preeclampsia. But the problem is that those studies never really looked at what happens if these people really have high amounts of depression or have a lot of symptoms pushing up that AVP. And that's what we hypothesized. So because we had all this data, we were able to kind of bin people according to how bad their depression symptoms are, by none to mild, moderate, and severe, by a standardized scoring. And what you can see here is that, in fact, if your depression symptoms are well controlled, that those who are SSRI treated actually have a lower rate of developing preeclampsia. And so that was the first time this had ever been shown. And so we asked the question, is it vasopressin related? Is it that thing in the brain that gets released in response to stress and blood pressure problems? And so we measured copeptin as a measure of vasopressin release. And we see that, in fact, in people who are SSRI unmedicated, you see an increase of copeptin in those who have preeclampsia. Not a surprise. And again, this is early on in pregnancy, less than 20 weeks. But if you look at the folks that have SSRIs on board, that in fact, overall, the vasopressin or the copeptin goes down. And more specifically, that copeptin is really down in preeclamptics. So what this tells us is that it's more than just a coincidence that we have these problems that are associated between preeclampsia and depression or anxiety or any of these neurodevelopmental things. We think that there's probably a real mechanism going on in the brain and in the placenta and in the body of the woman who is carrying this pregnancy. And so that's part of the work that we're doing. Some of the things that we've been able to do is this is one of the high impact articles that we published recently, along with my buddy Tom, looking at fetal circulating RNA. So that's a genetic code and trying to predict future health in pregnancy. So this is like the blood test you could get. Many women who are pregnant sometimes get the cell-free DNA test. When I talk to my patients about it, they always say, oh, that's the one that told me if I'm having a boy or girl. That's the same test, but it also looks at, whether it looked at your risk of Down syndrome. Well, this is a little bit of a different test, but very similar technology. And so because we have these samples, because we have this data, we're able to develop some new biomarkers. This is one analysis of looking at genetic signatures of gestational age. One thing that's hard for me and Tom is that we have patients that just kind of roll in into our labor and delivery, and we have no idea how far along they are. And ultrasounds are only so good at predicting how big this, how far along a woman is in gestational age. So we may actually have a marker for those, or a genetic signature for that. And of course, because of our obsession of preeclampsia, we've identified a very specific genetic signature that helps predict preeclampsia less than 20 weeks of gestation. We know that there's more than just what we're thinking about molecularly that helps with the care of those who have preeclampsia. And so as part of I'm one of the executive committee members for Iowa's Institute for Clinical and Translational Science. And in that role, I help run a very specific project to help improve postpartum care in preeclampsia. And so part of the problem is, as many of you already know, there's an increased risk of cardiovascular disease with preeclampsia. And so we need a better way to be able to monitor blood pressures postpartum. Sometimes these really fancy blood pressure cuffs that are Bluetoothable, they can be complicated in terms of making sure that they link to make sure we get the data into the system. We are studying in this project with the ICTS, what are the best ways to actually get blood pressures remotely? And one of the cool things we're doing is having patients take a picture of their blood pressure cuff, and then the software is able to automatically recognize the numbers and then shoot them into a database in which we can actually see all of these blood pressures real time. The other thing that we talked about earlier today is that there is a challenge in getting women to see their provider postpartum. As we've mentioned before, 50% of those who develop preeclampsia actually show up for their 6-week postpartum visit. And so instead of our patients coming to us, we're going to go to them. And so part of this project that we're doing is that no matter what, parents will bring their kids to their well child visit. And they will forego their own visit. And this is a true fact. I think Tom can back me up on this, that they will end up going to their pediatrics visit. So what we're doing now is trying to find, we're going to try to recruit women in these well child visits, and we'll have access to their medical records and and screen them for blood pressures that 1 week postpartum and even beyond that. And so we're going to them, and then we're going to try to recruit them to be part of a study where the blood pressures are going to be will be controlled by a network of pharmacists that we have through the state of Iowa. Because again, these people aren't coming back to us, but how do we go to them to provide that care? So that's part of the project that we're doing. Some of the other kind of biomarkers that we're able to come up with is because autism is such a big neurodevelopmental disease that's been associated with preeclampsia, we have shown that we actually have a very specific signature that we see in baby's cord blood that can predict autism 10 years down the line. And all of that is because of all of our samples, as well as the clinical research machinery that the bank affords. We've also done the same thing with our friends at End Preeclampsia, and this is part of this is the paper that we were referencing earlier, where we wanted to ask the question is, what is the effect of social media with regard to support and education for pregnant people? And the truth is that there are still holes in our care and our counseling for pregnant people. This was a mixed methods study where we had where Dr. Faro did 30 to 45-minute interviews with the moderators to identify really important themes. And these were the themes that we identified that were important, that they heard coming from the members: emotional, social support, mental health, education, personal health advocacy were major themes. And then we followed up with a quantitative survey of many of the members to ask what their experience was with regard to their care. And 68% of our survey participants clearly wanted to be more involved in the decisions about their care and treatment. And over 30%, this is what kind of got me, felt that they were not informed about the danger signals post-discharge after their preeclamptic event. And so that was very eye-opening to me. On top of that, we were able to on top of some of these kind of quantitative number type things, I think the most important thing that we got were some of these quotes that we've got from patients from the interviews. This is, I think, a really super important one, and I think that focuses on the PTSD and the trauma and the depression, anxiety that can come along with preeclampsia. We know it happens. The members know it happens. And I think it's important that we were able to put that out there, saying that how important it is for us to be addressing not only the risk of cardiovascular disease, but also these neuropsychiatric challenges that women who have preeclampsia can face. We also noticed too that we also asked, we're very fortunate that End Preeclampsia is a worldwide institution. And so we got to ask the question of, are there differences between US participants and non-US participants? And there's some really interesting things. This is just an example of one of the questions where it says, when you had important questions to ask your provider, did you get answers that you could understand? And it's I'm glad to see that the US participants get about 40% say yes, where in other areas, only 27% said yes to that question. So there's, if you want to have a comparison between what we're doing in the US versus other places, this is an interesting paper. So what did we learn today? Well, we know that between Donna and I, that pregnancy health advances have been limited due to a lack or of a decreased national research focus. And I hope you know and understand that pregnancy health really affects everybody. And certainly preeclampsia is a great example of that, how there are short and long-term effects of the mother, the child, and even the grandchild after that. And advances of preeclampsia care are happening everywhere and continue to build here at the University of Iowa. And with that, I'm happy to take any questions.
You packed a lot.
Sorry.
Presentation. No.
I'm from
apologies.
Chicago. I talk too fast.
Necessary. No, that was fantastic. Thank you for that. So when can we see a copeptin test?
That's a great, I wish I could tell you. That this is part of the reason why, it's the way things happen through the FDA and all that. There's testing and testing and testing and testing. And that's why it often takes, up to 10 to 15 years for a drug or a device to go get into the market. So we're still working on that.
Yeah. You often see like headlines, like, and they're usually very dramatic, like researchers have found a cure for preeclampsia. and then you read it and it's like a mouse study of like 10 mice or something, or, you know, something very small. And like, that's great that we're making those steps. But, you know, as a lay person, as a non-clinician or scientist, we could get our hopes up, right? For things
Right.
that are in the beginning stages, and it can feel frustrating to us that it seems like everything is in the beginning stages, but those stages can be many years long. But I think the important note is that it's happening, right?
Yes,
The
it's
research
happening.
is happening, and things are in development, and we're making strides in that way. And Santillan Lab is a leader in that arena, and so we appreciate that. And the fact that you and Donna also don't seem to sleep. I'm pretty sure you don't sleep with all of the papers that you guys churn out, and all the work you're doing. So we, the patients, appreciate all that you do for us. Let's see, there were a few questions. Let me go down and see which ones that may be appropriate for this particular future heart disease risk. What if your preeclampsia, or in her case, HELLP syndrome, occurred late? Does she still have the same cardiovascular disease risk? Are there varying levels of it?
So the way there are multiple studies, and so when we think about preeclampsia from a research standpoint, from a clinical research standpoint, there's two kinds: mild, severe, or early and late. So clearly there's plenty of data showing that the CVD risks are worse in those who have to be delivered early, because that is the marker of a more severe preeclampsia. It is also true that even if you're at 36 weeks and you have HELLP syndrome rather than just plain old mild preeclampsia, that your risks are elevated. Some really old data would seem to suggest that if you have HELLP syndrome, your risk of getting preeclampsia again and cardiovascular disease is like in the 50% range. So it's almost like flipping a coin. Now we know that it's actually less than that. The year-over-year risk of developing preeclampsia again is probably closer to 13 to 14%. Yeah.
So sorry to say, Lisa, knowing your story, get that cardiovascular workup, have a baseline with a cardiologist, make your annual well appointments, make sure your glucose, lipids, blood pressure, weight, diet, exercise, all of that is in check, whether that is through medication or your normal lifestyle. You can reduce your other risks. I guess we'll say reduce your other risks because you already have this one. So in talking about the stress component, she said, surely feeling stressed during pregnancy can't play that much of a role. Are we talking about general everyday stress, or are we talking about more impactful, stressful, social structure stresses?
I am unaware of those sort of studies. So the studies that I mentioned were kind of the impactful stress, like 6 months, somebody in my family died, or I was abused as a child, or a horrible divorce in my family, or something like that. That is, and those tend to be a little bit more dramatic, like coming to work for a university, or like all of our day-to-day life stress gets a little bit harder to quantify when we're trying to think of a good clinical study. And so unless you have a really good measure of day-to-day stress, and the type of measures and instruments that are out there to measure that stress are actually signs of more severe, clinically significant anxiety, depression, and what have you. So you're right. It's the data is out there is something like more dramatic sort of stress.
Like if you're living in a refugee camp and you're pregnant, and that kind of daily stress. Okay. Let's see, what else was there? Low PAP-A results, 12 weeks. Is that an indicator for increased risk of preeclampsia, or how
So,
accurate might it be?
when it comes to these soluble factors, it's a little bit all over the place. Not just PAP-A, but if you AFP was like the progenitor one, was the alpha fetoprotein, which we used to use. We used to help screen for spina bifida or open neural tube defects. A lot of that, we get a lot of those, and we always talk about there's something funky with the placenta if it's too high. PAP-A is something that other places use as sort of a screener, but it's certainly not, in and of itself, it's not the diagnostic test. It is not the troponin, troponin to heart attacks. There isn't a relatable thing.
Like low PAP-A is maybe one teeny tiny thing, and if it was combined with a few other things, that might give more cause. And I think the Europeans use it as part of a protocol, and it's one of several things that they look at. So on its own, not necessarily. Also, they think you're funny. So
funny looking, is that what you're saying?
No, funny. You're making Lisa laugh. So it's fantastic. I think that is all for those questions. Thank you so much. I think Jennifer Heiniger today talked about and as did Donna, about the research that's occurring, and then actually getting to see some of the actual research that is happening and what it can lead to. Can be, I think, very hopeful for our members, because we do kind of hear this theme or this story that, oh, no one's paying any attention. And while women's healthcare and maternal healthcare is underfunded significantly, it is happening. There is research happening, and we're making headway.
Can I make one comment that sort of pulls together both Mark and Donna's talks?
Sure.
Because I've been thinking for a long time about why are we having so much trouble finding biomarkers for preeclampsia, and why is it underfunded? And I think part of the systematic issue is that a lot of the way the scientific world thinks about biomarker research is based on cancer. That you find a marker that says you're at risk of cancer, and then everything else becomes obvious at that point. You get screened, you get therapy, you get surgery. And I think that paradigm is dominant, but I don't think it's serving us very well in pregnancy-related research. I think we need to move into a more unique paradigm of risk and management and stratification and therapy, and how do you handle the fact that somebody may be at risk, rather than this cancer paradigm. I haven't got it fully worked out. I'd be interested to see what Mark's thoughts or Donna's thoughts on this would be, but I think it is part of our headwinds, is that we just have a paradigm that we're borrowing from another field.
And I don't think the paradigm is wrong, in the sense that preeclampsia specifically is very heterogeneous, just like cancer, right? And so I think it's just cancer's got a significant head start in being able to understand what the molecular underpinnings are. And so to find that, and so we are the preeclampsia, you, me, the preeclampsia research enterprise, we are babies in that field. And only what was it, it's 2 years now that SFLT-PLGF was FDA approved. But even then, it's not saying that this is what the diagnosis is. What it's telling us is potentially you might have severe preeclampsia in 2 weeks. And so that's and with cancer, it's like, yep, you have a CA-125 that is super duper high. Yep, that's it. You got ovarian cancer or something. And so I think that's part of our challenge. And now, if the research enterprise actually starts focusing on women's health, and particularly preeclampsia, as we've talked about, then maybe we'll actually make some headwinds. And you're right, it's the headwind of please pay attention. There are more things than just cancer out there. So.
Great. Let's see. We may have a couple more questions. Yeah, Lisa just makes a point that, during her pregnancy, all of her focus was on the baby's health and not hers. And I think that's a common thread. And I think, most of us with wanted pregnancies would do whatever we needed to ensure the best outcome for our babies. And I think that ties up some of our patients and gets them into with worse outcomes because they think I'll just tough it out. I can just tough it out and I'll be fine. And I was one of those. I was like, oh, my blood pressure went down to normal range. It's fine. And they were like, well, you're fine now, but your baby's not fine. And so there can be fetal indicators as well. And it's not a matter of toughing it out. Again, you have to be healthy for that baby as well. So it's a great point, Lisa. Thank you, Donna, and thank you for bringing along Mark too. He's always helpful. We appreciate you letting him have a few minutes to talk to. I appreciate all of your time and all of your expertise, and I hope that our members really take something away. We'll probably have follow-up questions, so I will get those to you in email. If we can, as people are watching this and watching the recording later on, pop your questions and your feedback in the chat. We'll be following along and try to get you some answers when we can. This has been the best patient summit, very first patient summit one could ask for. And I appreciate all of you on the screen and Katie, who's behind her screen here, for making it happen, as well as our presenters and speakers from earlier in the day. So we will sign off now from the 2024 and Preeclampsia Patient Summit, and we look forward to doing this again next year.